Hi, welcome to the lecture on environmental adaptation of animals. Now we will reveal more mysteries on the muscle physiology. And the title of this video is Excitation, Contraction, Coupling, and it might be the most difficult part on the muscle physiology at least. But anyway, the excitation, contraction, coupling is just how the muscle cells are regulating the contractility. And we already know how the myofibrils are making the contraction. They need ATP and they need calcium. And the finding that calcium has some kind of importance for the contractility is nothing new. It was found by Signe Ringer in 1883. Adding calcium chloride in the solution makes these contractions larger. Okay, where the calcium can come from to the cell? Well, okay, you can have calcium channel on the cell membrane, so the calcium can come out from the cell. Then the other way is that you can have calcium store inside the cell where you can release it. And to reveal that which is which, if it comes out from the cell, then it has to be something to do with action potential. Because on the cell membrane we have calcium channels that are voltage gated. They are opened only at depolarized voltages. So that's why they can be active only during the action potential. And in the cardiac muscles, the calcium concentration rises quite fast when we are starting action potential, and it will be decayed at the same time as the action potential is ending. Over here, the action potential is on black, calcium concentration is on red, and the contractility is on green. There's not so much calcium coming through the channels, and that will get the calcium concentration lower because there's also some calcium pumps that are pumping calcium away from the cell or put it inside the cell in the storages. And then this contractility comes a little bit late because now you don't have, it's not only related on how much calcium you have, but it also takes some time to move the myosin and actin. But then if we are comparing the action potential of a skeletal muscle, it's only a very short peak. Only a few milliseconds. You can't even open the calcium channels during that time. Or if it's opened, there is not much calcium entering to the cell. And this contraction happens long after the calcium action potential has repolarized back to resting membrane potential. So it can't be regulated by extracellular calcium. So if we think on the striated muscle, what is the basic structure inside the cell? We all quite often think it as a box. Okay, it's a very long box, but the diameter is maybe 100 micrometers, whatever. And then we have calcium entering the cell and causing that, there is the contraction. But actually, the fine structure of the cell membrane is pretty much more difficult. There exist caveats like T-tubules and caveoles that are bringing the cell membrane close to the center of the cell. And some of these T-tubules are quite often interacting with calcium store, sarcoplasmic reticulum, SR. It's modulated from the endoplasmic reticulum in other kind of cells, but now because it's used for calcium store, so that's why it's called sarcoplasmic reticulum. And this helps then the calcium delivery deep in deeper parts of the cell. So although the cell diameter was 100 micrometer, so you still need some caveats like T-tubules to bring the calcium in the center. And if we are comparing how these T-tubules are different from the sarcolemma. So some ion channels are existing only in the T-tubules. For example, in cardiac myocyte, you can see there on the blue that, okay, the only voltage-gated calcium channel exists in the two T-tubules. So that's the only place where the calcium can enter in the cell. And even then, there might be differences in the sodium channels there in the red, or potassium channels in the green, etc. So some are existing in the cell surface on the sarcolemma, or they're in these T-tubules. And how it can be working, this EC coupling? Well, these skeletal muscles, they are not very spontaneous, thank God, but they are regulated by motor neurons. And these motor neurons are releasing acetylcholine on the neuromuscular junction. And when the acetylcholine is released, it will be binded in the acetylcholine receptor. This acetylcholine receptor is very nice. It's non-selective ion channel. It's very fast to open, but even very fast also to close. It's very difficult to measure that way. But because it's a non-selective ion channel, it permeates sodium, potassium, whatever that you have there. But because the extracellular solution is containing mostly sodium, so there's a high sodium influx on the sarcolemma. And this will depolarize the cell membrane and cause the action potential locally. But then the action potential starts to propagate in both directions. And one of the places where it's propagating, it's also propagating in these T-tubules with a little bit different ion channels. And when it goes deep inside the cell with the T-tubules, it will face difficult word dihydropyridine receptor. So it activates a receptor that is how this receptor is working. It's just measuring that, okay, now the voltage has been changed. And that triggers a calcium channel, which also has difficult word, a ryanodine receptor. But it's the calcium channel there on the sarcoplasmic reticulum. And this will lead that, okay, now the calcium is released from the acer, and that will facilitate the myosin-actin interaction. And why these receptors have so difficult names? It's because ages ago, before they were finding that, okay, what kind of proteins there exist, they noticed that, okay, one of some benefits or some action in the striated muscle we can see by having ryanodine or dihydropyridine. And because if you are releasing calcium, either from extracellular store or from intracellular store, you also need some pump to pump back the calcium back to the sarcoplasmic reticulum. So it's going there in the background. Well, how then to limit the contraction? Now we know that, okay, we can cause that, okay, there is a calcium is leaking from the sarcoplasmic reticulum to the cytosol, but we also need the limitation. Well, if the dihydropyridine receptor turns back to its resting conformation, that will close this ryanodine receptor. And that's why the release of calcium is transient. And the easiest way is that, okay, it happens during the repolarization. So, end of the action potential. We remember that, okay, action potential was only a few milliseconds in the skeletal muscle. So most probably, when the action potential has ended, now the dihydropyridine receptor is sensing that, okay, the membrane voltage has turned back to negative, and that will close the ryanodine receptor. But also, if it's very high calcium, it will also close the ryanodine receptor. Okay. Let's have all these things on a scale of the time. So we have first this sensing that, okay, the acetylcholine was released and sensed, and there is an action potential first locally, but then it will be propagated throughout the sarcolemma. And that takes maybe 5 milliseconds or even 10 milliseconds. But then, when it reaches the T-tubules, it takes less than 1 millisecond to propagate in the bottom of the T-tubule or to activate the ryanodine receptor. So these are very fast things. And then, if you are releasing calcium, it takes few milliseconds until the calcium reach the troponin, so it can start the tension. And then, most of this time is used for this contraction, because you need some physical work to turn the ATP for movement of proteins. So that takes tens of milliseconds before you can get the peak tension in the skeletal muscle. So it seems to be that these T-tubules and ryanodine receptor, they don't take much time on this contraction. They are the fastest way. Okay, but then we have calcium-induced calcium release. Because the dihydrogenide receptors, they are also calcium channels. They mediate calcium ions in the cytoplasm. So it could be that in some cases we could have operable contractile cells without the need of sarcoplasmic reticulum, because now we have entry for calcium. And in the striated muscles, this calcium that can go through these dihydropyridine receptors, it's inhibiting the receptor itself, but also activating the calcium channels in the sarcoplasmic reticulum, because the calcium concentration is still much less than when it's inhibiting this calcium release in ryanodine receptor. So then you have this calcium-induced calcium release. So there's calcium influx from the intracellular storages, and in the skeletal muscle, this is not the main mechanism, because the dihydropyridine receptor and the ryanodine receptor are bonded. But still, even in skeletal muscle, not all the ryanodine receptors are binded on these dihydropyridine receptors. And the interesting thing over here is that, okay, now we know that, okay, these both are calcium channels, but for some reason, people like to use these receptor names, although we could have that, okay, we have T-tubule calcium channel and sarcoplasmic reticulum calcium channel. Okay, we then might think that, okay, we might have some variance in this calcium release in regulation, because we have 3 different types of skeletal muscles. Oh, sorry, on muscle types. So, in the smooth muscles, the calcium for the contraction is delivered from the sarcolemma. So it doesn't need the endoplasmic reticulum or sarcoplasmic reticulum for the contraction at all. But in some cases, this calcium is released from sarcoplasmic reticulum to activate calcium-dependent potassium channels that are hyperpolarizing the cell membrane. So it means that, okay, if you are releasing calcium from the intracellular stores, you're actually making the cell rest. And quite similarly, we have in the neurons, because over there we can have extracellular source for calcium entry, but then it's affecting on the release of calcium from the endoplasmic reticulum and activate the potassium channels. But then in these striated muscles, we either have, like in the cardiac cells, that, okay, you have calcium entry through the cell membrane, and this will cause calcium-induced calcium release from the endoplasmic reticulum, from the sarcoplasmic reticulum, and then you have the contraction. Or you might have direct link between these 2 channels, and then you are having calcium only from the intracellular storages. So let's have one more updated figure on this excitation contraction coupling in the skeletal muscles and heart. So in the skeletal muscle, these two channels are binded together usually, and they will cause the release of calcium from the intracellular stores. There might be some calcium channels that are not binding to ryanodine receptor in the T-tubules, but usually their role in the skeletal muscle is very limited. And then there exists also one stabilizing protein for this ryanodine receptor, and that's regulated by beta-adrenergic regulation. So there's a G protein and cyclic AMP, etc. But beside this contraction, even in skeletal muscle, this calcium release can have some side effects. So first of all, high calcium can regulate mitochondria to produce reactive oxygen species. But also it can affect by calpain on the dystrophy of the cells, etc. But then in the heart, there's no mechanical link between the calcium channel in the T-tubules and the calcium channel in the endoplasmic reticulum, sarcoplasmic reticulum. So there's this calcium release, calcium-induced calcium release. And it can be affected also by beta-adrenergic receptor, by cyclic AMP, by this stabilizing protein. But in the heart, there exists also another receptor that is releasing calcium. Is this inositol triphosphate receptor. So we have this other G protein mediated receptor that is causing that, okay, you have phospholipase C, and then you produce the IP3 from PIP2, and then you are releasing calcium from the, over here you can see that, okay, other end of the sarcoplasmic reticulum. But when you have high calcium, you can have it again problems in the reactive oxygen species in the mitochondria, but they can also affect on the hypertrophy and aging of the cell. And then on the smooth muscles, they come a little bit differently. So now the calcium is delivered by the sarcolemma. There is a voltage-gated calcium channel or the sodium-calcium exchanger. But vasopressin is releasing calcium from the endoplasmic reticulum in the smooth muscle. So there can be, in some cases, also these intracellular stores. And then this calcium is binding on calmodulin and activating myosin light chain kinase, which will phosphorylate the myosin light chain and cause the contraction. But at the same time, there is a pathway that is affecting on the myosin light chain phosphatase that removes the phosphate and causes this relaxation. So you have pretty much different way to contract in the smooth muscle and the skeletal muscle. And let's compare these. So on the left hand side you have the striated muscle, on the right hand side you have smooth muscle. So in the striated muscle we have calcium is binding on troponin C and exposing myosin binding sites. On the smooth muscle it's modulating on calcium calmodulin. Then the ATP is binding on myosin, but over here it's working as calmodulin, myosin light chain kinase that is using phosphate to phosphorylate myosin light chain. And then in the striated muscle, ATP is hydrolyzed, myosin is in cocked state, and there's a cross bridge of myosin that being a new position on action. But over here, the phosphorylated myosin light chain is interacting with actin. And that causes the contraction. And then you have the whole circle on the striated muscle as well as a smooth muscle. But you have seen that, okay, it seems to be quite different. Thank you.